that, so it's suggested to use a photobioreactor system as an inoculum. So we always have a better monoculture compared to a mixed culture. So that's one of the suggestions here. So when this wastewater treatment using algae started, the history of the commercial use of algae culture spans about 75 years, with application to wastewater treatment and mass production of different strains, but in different countries. So the developed countries are already using this technology, but this technology is slowly emerging in developing countries. Yeah, the countries like Australia, US, Mexico are already using this technology in a big fashion. So we are also involved in bigger projects in Australia. We'll discuss about that. So the biotreatment with the microalgae is particularly attractive because of their photosynthetic capabilities, converting solar energy into useful biomasses, and incorporating nutrients such as nitrogen and phosphorus, causing eutrophication. So that's an advantage. So that's a major advantage because it's like a plant. We can able to use it as a big source to convert wastewater into value-added stuff. Algae can be used in wastewater treatment for a range of purposes, some of which are used for the removal of coliforms. If you have seen the reports or test analysis of different effluents, you can see that coliform bacteria in that. Okay, so the coliform bacteria and algae are enemies. If coliform bacteria dominates, then algae growth slightly decreases. If algae overcomes that and grows faster than it, then it suppresses the coliform bacteria. Therefore, it's always like enmity between these two people. Therefore, that's one of the reason I told you in the beginning. Once you have the wastewater treatment plan, you should have a proper analysis report of that wastewater to understand what are all in it. If it is more coliform, we need to select appropriate algae to handle it. If it is less coliform, inappropriate algae. So this is very crucial parameter. So we need to understand. So the reduction of both chemical and biochemical oxygen demand is highly possible, and it is a proven concept while using the algae as a wastewater treatment medium. So the removal of nitrate and phosphate are seen in different wastewater treatment processes, and especially it is very good in the removal of heavy metals, even iron. So you can see these pictures at the right. So that's what most of the rivers back in India. And I think the picture in the bottom is maybe Ganges or from Coombe River. So that's how it looks like. It's possible we can able to make it as a clear water, but it's a long-term project. So it's keep on increasing because of livelihood living in the sides of the rivers and banks of the rivers and the lakes. So it's giving more problem to the water, but unless or until we have a heavy fines or legislations to control it, it's not going to help us. But always prevention is better than cure. Here I suggest the same thing, prevention is better, but once it gets contaminated, yeah, algae is there. We can able to handle it, completely clear it, make it as a clean drinking water. That's possible. That's the near future. We're going to see some clear water. So as I told you in the previous slide, the coliform is a very important parameter in selecting an appropriate algae with the deals with the coliform, because all effluent has it. I have given some examples here. So the Bacillus, the total coliform, fecal coliform, and E. coli. So all these coliforms can be significantly controlled by algae. We have proven results for that, especially in the sewage treatment. We can able to handle that using appropriate algae, especially in the sewage treatment. So the reduction of both chemical and biochemical oxygen demand, the report says that 68% and 67% of BOD and COD are reduced while using algae in the biological wastewater treatment process. That's a significant number. So this gives more advantage for algae to handle the wastewater treatment process very effectively. I can share some results of our experiments in next couple of slides, which shows the reduction in terms of numbers, because it's always good to talk with the numbers. So removal of nitrate and phosphate. Yeah, this Chlorella vulgaris, the species I am talking about, it is a freshwater algae. We also have a saltwater algae, which is called as Chlorella salina, which we are working on. So this algae has the capacity to reduce the nutrient up to 86% inorganic nitrate and 78% inorganic phosphate. So we have proven records for that. So when you reduce that, then definitely we can significantly control the eutrophication happening in the sea. So it's possible at the industrial wastewater treatment site, if we have that facility to use the algae treatment to reduce the N and P, then we can stop killing the fishes in the sea and the river. Heavy metal removal from wastewater, as I told you, iron, we can able to significantly remove, and also the lead we can able to remove significantly to a below detection limit. We have some proven algae out of that 100 algae we have in our current culture collection. So that's also possible. That's the major advantage. That's only possible if we have a microalgae related treatment. Other processes, it's pretty hard to remove the heavy metals, but here it's possible. So this is the process flow diagram, which was developed by one of my students who was working for a dye industry to treat their effluent in an effective way. So as I told, the student wanted to focus more on the primary treatment. Between the primary and secondary treatment, he wanted to incorporate the algal technology to treat the effluent, but it is again a failure because of the more concentrated products present in that primary treatment step. Therefore, it is a tertiary treatment was suggested for this company. So where the dye effluent, secondary treated dry effluent was sent into the clariflocculator and then into a photobioreactor. So he wanted to control the contamination in that. Therefore, he went into the closed system where it has light, which gives more light energy to it instead of going into a sunlight, and also a controlled environment full of glass. It's pretty hard for other contaminants to grow and cause some problems. And going to the sedimentation, where he added the flocculant to remove the algae, and the remaining water, which is the supernatant water, goes out of it and used in different processes. So the algae which is coming out of the flocculated tank can be sent into the filtration to remove more water and then take out the dried product. So either you can use a tray dryer or you can use a spray dryer. Whereas if you are using a spray dryer, the solid percentage should be around 15% to 20%. In case of tray dryer, it's not a matter. It's like an oven. So that has to be taken care. So this specific flow diagram process was developed by one of my students back in India. So this table shows about the bacteria algae pond, high rate algae pond, and integrated pond. The different parameters are listed on the left. So what is the normal depth they are using in bacterial algae pond is 2.5 meter, whereas in algae it's just 0.2. It's a shallow pond. It's more shallow pond. Whereas in integrated pond, it is 4 to 5 meter. The salinity, it is increasing. So whereas in terms of high rate algae pond, it's not, because we are frequently topping up with water. We are controlling the evaporation rate as well. The retention time is always lower because the oxygen level is high in algae ponds. Okay. The land requirement is low because we require a small size pond to handle this effluent treatment. Odor problem, I told you already. So whenever bacteria comes into the play, we have a very bad smell. So here algae never cause such equivalent foul smells. So loss of nitrogen to atmosphere is not at all possible because it completely consumes nitrate and it grows. That's a major nutrient for its growth. So maintenance cost is very low and compared to other processes. It's like agriculture, maintenance is very less. So it's obviously it's very less. So phosphate sedimentation definitely not occur. It takes phosphate as well. It's another second important nutrient for algae. Time required for treatment is a very few days. The reason, the multiplication rate is very high. So it doubles, triples in a matter of 12 to 24 hours. Energy requirement is low. However, it depends upon the type of mixing you are using. If it's a paddle wheel, then there is a little bit of energy in it. It's for a big pond. If it is just airlift kind of mixing, then the energy is lower. So it depends upon the type of mixing you are going to do it. Efficiency quality is always high because it's the best method, best source to produce more good water from the bad water. BOD removal, it produces more oxygen, so it's good. Suspended solid removal, it's possible to a good extent. Harvesting cost is high. Again, it depends upon the process you are selecting. If you are using a simple flocculation method to harvest, then it's a low cost. If you're going for ultrafiltration, microfiltration method, what we are using here, then it is a high cost. So you need to decide based on the budget for that project. So I'm going to talk about the numbers, which I told you in a couple of slides back. These numbers are from dye industry wastewater analysis before and after it was treated using a particular algae called Senedesmus species. You can see the TDS reduced not significantly, but to a certain extent. The nitrate load is reduced to nearly to the half, the phosphate nearly to the half, COD significant reduction, BOD is the major reduction, TSS no, and then chlorides not as significant, but there is a reduction, the pH improvement and sulfate. This is one of the project done by my student back in India who developed the process for dye industry effluent treatment using an algae. This algae is a very good friend handling nicely the dye industry effluent. So the same project with the Chlorella vulgaris, it shows a quite a bit different result, but still you can see that the differences in reduction. The TDS, little bit, nitrate, half, phosphate, little bit, but not much like a previous case. Then the COD, not much like the previous case, but still there is a reduction. BOD, again, not much like the previous case, but significant reduction. TSS, yes, significant. Chloride, sulfite, a little bit, and pH improvement near to the neutral. So this algae is not preferable one for that particular effluent, whereas the Senedesmus is looking very soundful for that effluent, especially for the industry. Okay, I can share some pictures now. So this is a experiment of another project where the Chlorella vulgaris was used in the dairy effluent treatment, where you can see the dairy effluent is subjected to the algae growth. The algae is growing nicely inside. They give artificial carbon dioxide and the air mixing. And then this picture is very attractive. I told you we even tasted this clear water. It's just like nearly like a drinking water. So the effluent was like this. It was treated with the Chlorella vulgaris, and after the treatment become like that. So we just removed the biomass, I mean the algae, after it grown to a certain cell density, and then use a flocculation method to remove the algae. So this is the pure water, nearly to the drinking quality. So these are all the data which were given by the industrial partner for the dairy industry wastewater we were handling. I don't have the data for this water because this water nearly matched the drinking quality standards. So I don't want to put that here. You can clearly understand the difference between these two, except little bit of taste in that, which is not soothing taste, but other than that, it's nearly equivalent to the drinking water standards. Yeah, that's another example. Algae was utilized in treating a dairy industry effluent. Okay, so this next topic is about coal seam gas water. This is the project what we are currently doing in Australia, because many of you know Australia has more coal mines here. Therefore, more focus is on coal industries here. The major job market in Australia is coal industries. So these people approached us a couple of months back, stating that they have a big problem with respect to the pollution regulations in Australia. The problem is they are getting more water out of their bore. It is essential because they need to push the water into the bore to the gas out. So that water becomes a hassle for them to handle. The reason see. Therefore, they wanted to just get rid of that water, but they are not interested on the product what we can get from that water. Which means we suggested them we have certain algae which can grow in different salinity that can produce value-added compounds, but they are not interested in the product, but they are interested just use water. We can give the water free of cost. We can give the flue gases generated from our processes free of cost. Just get rid of the water. And they also gave an option of water in lorries or containers. They can just transfer it through piping system to the nearby land. So that land is a non-arable land. So it's a big project. So they, before going to that project, they just wanted to understand which area will be suitable for their salinity. They have different variety of salt water in their process. Number one is having a salinity of 25 parts per thousand. Okay. The number two, 320 parts per thousand, nearly to that range. Okay. So they gave, we want to get rid of this 25. Also, we want to get rid of this 300 plus. So when we were doing the research, we found out two algae are suitable for that. The number one is the spirulina, which can able to handle less salinity. Number two is the Dionella, which can handle high salinity. Therefore, we did a significant research and supported them with the reports. This project in a bigger scale. Okay, this is the big problem currently that Australia is facing with respect to the CSG people. One side the coal is mined, and the other side the water is not able to be used in any process except algae. So after successful completion of the first stage of this project, we are just moving into the second stage where we have plans to analyze the carotenoid-rich algae. Both has high value-added advantage. So at the same time, we can produce pure water which can be used in the processes again and again. That's possible. So yeah, we are working on that. This is the coal seam gas. So the proposal what we gave is shown in this simple picture. The left side picture talks about high salinity water. The right side picture talks about the low salinity water. So the coal seam gas industry will also supply the flue gases coming out of the industry. So therefore, when we expand this project into in a farm near to their facility, so they will be supplying their water, and that will be stored in a big storage tanks, followed by disc filter of 100 micrometer to remove the suspended materials, and then it goes into the ultrafiltration system to get more pure water. This ultrafiltration system we already are using with 0.25 micrometer pore size in that, which means we completely remove everything in that and then takes the pure water into a filtered water storage facility. That water is used for the cultivation of it. Okay, so this system can also be used for harvesting purpose. Okay, what happens? The concentration of solids in the raceway pond where the algae is growing is 0.05%. So we want to increase that at least to 5% using the ultrafiltration system. Again, we need to further increase it using a centrifuge system up to 25%. So, and then it goes to the tray dryer. In case, if you are going to the spray dryer, the product which is coming out of the ultrafiltration system is very dilute, 5%. We need to convert that into 15%. We have an additional here where we use a microfiltration system to convert that further increase in the solid percentage and send it directly to the spray dryer. And then if the people are interested in the product, they can go into the extraction mode using supercritical system. If not, they can directly go into the aquaculture feed or animal feed. This is especially for Duodenala salina, which grows in higher salinity. Whereas the right side, it is similar, but we are using spirulina for the cultivation because it can grow maximum of 25 ppt. For animals and aquaculture. So the processes are similar, but instead of ultrafilter, we are using a drum filter because of the size of the spirulina. Okay. Here, the Duodenala size is smaller, the spirulina size is bigger. So other than that, the processes are more or less similar. So here we are extracting to extract a phycocyanin and a protein, whereas here the possibility to extract carotene as a value-added source. So this is what we are going to do in a big farm, which is planned for 2021 for using the CSU water. This is possible for any effluent, but you need to characterize that effluent before deciding on the process. So this table will give you some glimpses about the CSU water. The first column, this column shows about the 90 ppt water which was given by the CSG industry to us. When you do the major elemental analysis, we came to know it has no nitrate. Other than that, it has a good amount of phosphate, sodium, magnesium, potassium, and calcium. You need to add anything extra algae cultivation except nitrate. Yeah, it's not at all present in the CSU water. We added this, and the total nutrient concentration is increased to some higher number. And then this is the result we got end of the algae harvest. So therefore, you can see that significant reduction in the sodium level, which means the salinity was significantly reduced. The magnesium and the potassium, calcium, the pH, everything came to active project proposal for expand this project in a bigger scale. So this is especially for Duodenala salina. I also have the reports for the spirulina plant disease. So it is similar to Duodenala, but the only major difference is it's using more salt into it. Rather, it is trying to accumulate its cell. So anyway, we are not much concerned about the sodium, but when compared to other stuffs, there is a significant reduction in nitrate, phosphate, magnesium, potassium, calcium, and also the pH. The pH, it is 7.9 because the spirulina can handle higher pH. So whereas in Duodenala, we dropped it to 7, be able to control it. So this is a very interesting project currently going on, and it is also attracting more PhD students and postdoctoral fellows into this market in different universities. Okay, so let's think about how to make consultancies and how to make this cultivation and harvesting of algae wastewater as a business model. Okay, so let's make some money for ourselves and also for the institution where we are working for. So there are many ways to get yourself involved in the algae industry. Algae industry can be very complicated and diverse, but with a little bit of expertise and training, you can lead the industry. So choose a correct startup point for that. So algae reproduce very quickly and need only sunlight or another form of energy, sugar, etc., water, carbon dioxide, and few inorganic nutrients. Okay. So what are the steps in forming a company, and especially algae-based wastewater related cultivation system? Just you need to first analyze the locality of the effluent. If the locality is possible to construct a farm near to it, then it has to be considered. Otherwise, the logistics is going to be a significant problem for transporting the effluent for your algae farm. Okay, so once you identified the location, and also once you assessed the quality of the water and the physiochemical parameters of the water, then you can decide on the number of ponds and scale-up activities. So number one, construct small ponds initially as per the required number, and then go into the bigger pond. Next is the site preparation. So this is exactly the work what we are doing here, how we created our site, how we selected a land near to the facility, and how our site was constructed. This happened a couple of months back, and we are now in a nice stage where we are going into the production mode. This is a part of University of Queensland's pilot level research facility in Brisbane. So the picture shows the next level of operation, where the site was prepared nicely, because we are trying to avoid using more energy for running the farm. So for example, if you have a nice elevation for your storage tanks, you do not require additional pump to push it from top to bottom. Just by gravity, it will flow down the water. So those kind of designs we did, and excavations we did, and this is a picture which shows that, so about the construction of the facility. So once that is done, then we found a nice place to make that water tanks to sit, each tanks of size 50,000 liter capacity. They are in a higher elevation, up to 2.5 meters from the ground level, from the pond level, and the CSG water, the other effluent water, can directly come into this tank, and we can use it straight away into our process. The next level, the construction of the bigger pond with the pond liners. So this is the pond liner into it. So you no need to make a complete concrete structure here. So just the sand, over that we have the pond liners. The sides are with rocks. You don't need to have a hi-fi system to build this. Just with available sources, you can do it. And then fixing the paddle wheel. The selection of paddle wheel is very important in algae treatments. So this is the best one so far, based on our research. This type of paddling with 6 paddles in it is so efficient in mixing the algae culture. It's completely, that paddle is completely stainless steel 316, because we are going to use high salinity water to prevent corrosion. We used a stainless steel material. And then once the bigger pond is in operation, we need to fill that with algae inoculum. How to do that? So the pictures which are shown on the left, the starter culture of our algae, and then it is scaled up in the laboratory in 250 ml to 1 liter flask. Then it goes to the 20 liter bag, then goes to the 200 liter bag, then goes into the 2000 liter small pond, and then goes into the 20,000 liter big pond before it goes to the 200,000 liter pond. So our maximum capacity of our bigger pond is 200,000 liter. So you can see that number increased by 10 folds. That's what the inoculum size is. So 20 to 200, 200 to 2000, 2000 to 20,000, 20,000 to 200,000. That's how it's a procedure to maintain a good inoculum size. This is the next step in the scale up. So here you can see once we have a nice cell density in the 20,000 liter pond, we transfer that algae into the 200,000 liter pond. You can see that carbon dioxide sparges, air sparges, and all sensors here attached to this pond. We can monitor it in a live feed, the pH, temperature, the turbidity, the salinity, and light intensity. Everything can be measured, and you can see it from your home how it is performing. So this, I wanted to touch a little bit about the mixing pattern. I told you this is a paddle wheel, one good type of mixing, which takes more energy. The other way you can use the mixing is by using the airlift kind of mechanism. where we have a V-shaped tank in between the pond. We pass the air into this area, and then the air which is escaping out of the other side will pushes the water to flow. That gives a good kind of mixing. But this kind of system is suitable for a smaller pond. For a bigger pond, you need to employ such a similar system, maybe 3 for a 200,000 liter facility. This requires a little bit of less energy when compared to the paddle wheel, which is cost effective. So design is rendered by our team, and we tested that in a smaller system. It's working good. A larger system, it requires at least 3 numbers to make it a good efficient mixing. So you can see the fully operational 200,000 liter pond here. So which uses the coal seam gas water to grow the algae in it. It's completely green. It grows like anything. It's when compared to a laboratory scale, if you see that in the real scale, it grows very faster. The optical density reaches very faster. So it's which means it is like 3x, 4x times faster growth than a normal cultivable algae. So what happens next once we have enough algae grown in the pond? We have 2 options. One is cost-effective option to do a flocculation to separate the algae, and then use the clear water back into the pond to cultivate again. So that's a cost-effective option, and also it is suggested for fragile algae like Dionella salina. So if this algae is going into the microfiltration or ultrafiltration, then the cell walls will break and it release the products outside. So we don't want to do that. Therefore, for fragile algae, we always follow the flocculation method. So we developed organically sustainable flocculant in collaboration with another company in India. So where we could able to use very minimum quantity of the flocculant. For example, 0.1 ml for excuse me 1 liter of algae water to flocculate it. The reason we wanted to use this is completely organic, which is produced from moringa seeds, neem oil, and also polyethylene glycol, which is used as a solvent here to dissolve these components in it. So it is approved as an organic flocculant. Therefore, when you are selling the product to the customers, you can still say we did not use any other inorganic flocculants. Therefore, the product becomes a food quality product. Either it is a feed for animal or food for human. It is approved as a food product because of the flocculant what we are using. That's recently applied for a patent, and its patent is pending for that complete flocculation process. So this is specifically for a fragile algae. If it is non-fragile, so then we go into our Licoflux. Licoflux is a equipment we got from Netherlands from a company named Turbine. So they are the world leaders in producing this particular equipment, and we frequently send report to them about the performance of this machine and how to alter that machine in the future to sort out other problems what we are facing it. So which means we are the brand ambassadors for this particular company, which is doing extraordinary membrane technology to especially for algae harvesting process. So it goes to the Licoflux, where we concentrate the algae up to 5 to 6%. And then it goes into the next system called the Licovibro, where it has 3 stacks of different sized, 4-sized filters. Here you have 0.25 micrometer, whereas here it is 0.2, 0.8, and 0.6 micrometer 4-size filters. The uniqueness is this whole system can vibrate like a sieve shaker. The vibrate, and it removes most amount of water, and we can concentrate the algae up to 20% solids maximum. Okay, so and then it goes to the drying. So either you can do it by a sustainable solar drying, just to spread it here. This is in our farm. Spread it here and then make it as a dried product, and crush it and make it as a powder. That's one of the company's product. This is also my company in Australia, the Algae Australia. So these products are belonging to them. So the process is the similar, yeah. Or if you want more refined powder, so this can go straight away into a spray dryer because it's less than 20% solids. So you can spray dry it and make it as a powder rather than going into the solar drying. So this is the satellite view of the facility, what we are currently operating. So where you can see the different sizes of ponds. This is a 20,000 liter pond, and this is a 200,000 liter pond. And it's a nice location where you have the river also. So it's called Brisbane River. So you can take the water if needed, or you can dump the treated good water back into the river if needed. Otherwise, you can recirculate the water into the same system. It's highly sustainable. CapEx, OpEx, these numbers are in dollar value, in Australian dollars. So if somebody is interested to make it as a business model and submit to a government, then I can help you in terms of Indian costing. It will be very significantly less money when compared to Australian costing. So this table is formulated based on the Australian economy. So the process is divided into cultivation, dewatering, drying. If you are interested in the value-added product, then it comes into the fact. So here the value-added product is oil. So the amount of carbon dioxide, solar, labor, maintenance, total, amortization of the capex. So all it comes to a less amount here you can see. So to produce 1 liter of bio oil from algae, you're just going to invest $2.39. A dollar is equivalent to 50 rupees, Indian rupees. So which means, you know, the value of oil in the bio oil. I'm not talking about the biodiesel. This is the omega oil, omega-3, omega-7 oil. 1 liter is going up to 270 to 300 US dollars in the market. Therefore, you are not only taking care of the effluent treatment, wastewater treatment, and also you are producing the high value product, which can be sold in the market for higher rate. You get a high margin. Therefore, this capex OpEx is very specific for the oil-based product. We have a similar capex for protein and the carotenoids. So this table shows the capital investment and the energy cost associated per year. Yeah, these numbers will help you if you're really interested in some consultancy opportunities in the future. So what is the take-home message? We are in the end of the presentation nearly. So a take-home message: algae have become significant organism for biological purification of wastewater, since they are able to accumulate plant nutrients, heavy metals, pesticides, organic and inorganic toxic substances, and including radioactive matters in the cell bodies with their bioaccumulation ability. So that's one of the take-home message I would like to tell to everyone who are listening to this lecture. The removal rates of particularly high-rate algal ponds are almost similar to the conventional treatment method, but it is more efficient with lower retention time. Yeah, that is very important. When you reduce the retention time, then it's a big cost-saving approach. Value-added products, so yeah, more than treating the effluent, we have a big market for value-added product. So the effluent is coming for free of cost. The flue gas is coming for free of cost. Your investment is very little, whereas you're going to make big money out of that. So that's a big advantage of this process. That's a take-home message. Okay, so for possible collaborations, the person in this picture is Professor Pierre Schenk. Is my team leader here in the University of Queensland. He's a well-renowned algae and microbial scientist in the globe. So we are very much interested for any bilateral projects between India and Australia. You can contact me and I can forward that to Professor Pierre, and we can have some meetings, and whenever there is a call, we can work together and make some sustainable projects happening in India and Australia. So there is also possibility for universities to create a program like Manipal Institute of Technology. There is a program in Manipal Institute where the student can spend 2 years in India, and then the remaining 2 years in Australia in University of Queensland. Such kind of collaborations are also possible. So we need to work it out if your institute is really interested in that. But with the current scenario, With the COVID situation, it's not possible until until July 2021. The Australian borders are closed, but the paperwork can be done before that and make some progress on that. Okay, that's about university collaboration. So the next one is the Cuponics, the company where I'm working as a CTO here. So we also would like to be a part of industrial partners, industrial collaborators with any bilateral projects. So whenever UQ is applying for any algae related, Cuponics will automatically join them as industrial partner to put some value added benefits to that project. That's possible. The other company, which is my own company, Algae Australia, is also interested to do some consultancy projects here. So that's also a possible collaboration. So the two companies listed on the right side, the Seagrass Technologies and the Glacos Algae, are based in India. The Seagrass is in Pondicherry State in Karaikal, and the Glacos Algae is in Nellore. It's in the creation phase now. So what can be done is the Seagrass technology is recently applied for a DSIR recognition, which means the university people who are registered in university or engineering college, people who are registered for their PhD can do their research work in our facility here. So we have a pilot facility dedicated for research activities pertaining to algae, especially effluent area or sustainable route. That's possible, highly possible. We're also signing a lot of MOUs with the different universities in India, so which makes the students to undergo industrial training in the algae sector, and also interested people to get some hands-on experience how to cultivate, harvest, and pack algae products in a commercial kind of environment. So we are also giving that training. So that's also possible. MOU is possible, and PhD research because of DSIR recognition is possible, and student exchange is possible. And the last thing is you can still do some consultancy activity by using our technologies there. That's possible. So it is again possible with the Glacos algae, but it is currently in construction mode. So once this company is created, so this company will be the Asia's largest algae cultivators in a sustainable route. So that's what we are aiming for. So that's going to happen in 2021 because of delays caused by the COVID. So these are all possible collaborations you can think about, because I know everybody is from different universities and colleges. Definitely it's going to give some mark for NBA. So think about that. Thank you. And you can see a funny picture of me here. That shows how I'm fond of this algae. This is my car. This is my nameplate. This is the nameplate of my car. It shows algae. So it's easy to get such nameplates in your own fashion here. So Algae Australia, it shows Algae Australia. I have provided my email IDs here. That's a Cuponics ID, and this is the personal ID. This is my WhatsApp and the mobile numbers, anytime reachable. Just bear in mind, I'm 4.5 hours ahead of your Indian time. So I'm reachable easily. Yeah. So thank you for listening. I think I'm still in the time, so we can have some questions for the rest of the time.
Yeah, thank you very much, Professor Swaminathan, for a wonderful session. Am I audible?
Yes, madam. Thanks.
Yeah. Excellent presentation, Swaminathan sir. You have given insight into what is happening in algae in Australia as well as in India. I have some questions to ask. Before that, I'll request the participants to interact with Professor Swaminathan. He has shared his mail ID also for your further research and other queries. You can also contact him through his mail ID. So if there are, we can take 2 or 3 questions from the participants. Participants, please. Sir, Swaminathan sir, shall I ask, I have certain doubts with you to be clarified. Shall I ask you some questions before the participants?
Yes, sure.
Yeah, you told about the COVID and related to COVID medicines related to algae. You mentioned,
Yes,
can you just elaborate, because we are much interested to get medicines for the COVID, is it? So it has not yet been reached India. People are suffering, and much deaths are there in India comparing to the other countries. So we are very much afraid of when the medicines will come. Can you elaborate? Is there any successful work is going on in related to algae in COVID treatment?
Yes. So the University of Queensland is the one of the top universities which is currently working on vaccine development. So that's a different people in the same University of Queensland's School of Chemistry and Molecular Biology. So, but our focus is to prevent. Their focus is to prevent using a vaccine. Our focus is to treat the COVID affected patients using this algae technology. The algae which is used is Nanochloropsis oceanica. So this algae produces omega-3, omega-7 and other value-added fatty acids. So these fatty acids we have tested in our one of the research in animal models for different viruses, where we could able to improve the immunity of the cells and protect the cells from this kind of viral attack, especially the lung cells. So this anti-inflammatory properties helps a lot to get protected from lung diseases, which is a major target for COVID infection. So therefore, we wanted to do this experiment with the animal model with the COVID virus. However, there is no reports available so far to use it in a animal model. Therefore, we had a discussion with our scientists in different sectors and came to understand there is a genetically modified animal which can be used for this kind of project to test the anti-inflammatory compound secreted by the algae in its cell to understand how efficacy it is developing, how good it is in treating COVID. So that's what we proposed to the federal government. At any time, we are expecting a successful grant, and we are closely working on it.
Sir, I'm going to unmute the participants. They may ask questions.
Yeah, please.
There are questions in the chat box. Now I have unmuted the participants. Participants can unmute themselves and speak.
Very good afternoon, sir. So really your presentation is very informative. And
Thank
I
you,
wanted
sir.
to ask one question. So when we use flue gas for taking carbon dioxide from flue gas, so if you admit the flue gas to the algae, flue gas temperature is always more than 100 degrees centigrade. So when you admit to the algae will not alive position. If you reduce the temperature in the flue gas, the carbon dioxide convert to carbon monoxide due to the reduction. So what way you can allow the flue gas to the carbon dioxide, how to take to the algae by means of directly through the flue gas?
Yeah, that's a good question, sir. So which university you're from?
So I am the same department. I am Dr. Stalin. I am one of the co-coordinator.
Okay, nice to meet you. Yeah, that's a good question. So we are using a technology here called Arthena technology. It's a patented technology created by CSIRO. CSIRO is similar to CSIR in India. Here they call it as a CSIRO. It's a similar kind of expansion. So they developed the technology where they can able to capture these flue gases and separate just the carbon dioxide and then store it in the container and then send it to the algae. So that's the technology we are currently exploring. So as you told, we cannot use the raw flue gases straight away, but we also did some research where there are certain algae which can still consume carbon monoxide as well. Though it is toxic for certain organisms, there are certain algae which can consume carbon monoxide along with the carbon dioxide. So that's also possible. So therefore, these two approaches can help us to use the flue gases in an efficient way without creating any problem.
Can we use any type of flue gas to the algae?
Especially if it is having hydrogen sulfide, no, we can't use it, hydrogen sulfide, but carbon dioxide, carbon monoxide, NOx, SOx can be used.
Okay, thank you. Thank you, Dr.
There is a question from Kavya Siddeshwar. Kavya, madam, you can ask your question to the resource person.
Yes,
Kavya, madam is here.
yes, ma'am. Yes, ma'am.
Yeah.
Good afternoon, sir.
Good afternoon, madam.
Thank you for the fruitful session, sir. So my question is, can the leachate
Can
from
you
the labs?
introduce, madam? Kavya, madam, you please introduce. Where are you from?
Yes, ma'am. I'm a research scholar from Bharathidasan University.
Okay.
Sir, can the leachate can be treated with the help of algae coming from the landfills?
Yeah, it's possible as long as the physiochemical parameters are suitable for algae. So that's what I told. Any effluent, any waste can be effectively treated by algae as long as we have the data in hand. If we have a nice picture in on hand, we can decide. Okay, this data. Okay, we can select this algae. Such a kind of library we have. If we have the data, you can take something from the library. I mean, I meant library as the algae culture. So we can give it and then try a test. It's possible.
Yes, sir. So my next question is, when the treated water from the algae, when it is used in the aquaculture, so won't there be any sort of biomagnification, or it will be toxins will be in less like a negligible amount?
Yeah, it is in a BDL, so below detection limit, because we are nearly converting the water into ultra pure water. That is the purpose we are using the UF and other systems. The algae itself will take all the TDS, most of the TDS. So therefore, even the TSS will be significantly removed. Also, algae takes care of the heavy metals. Therefore, it's going to be a excellent source to use in the aquaculture industries.
Last question, so how can we treat like algal blooms? So when there is oil spills in the sea or something, they see the algal blooms have occurred. So how can we treat those, or how can we?
Yeah, that's the recent project we are working with Klamath Botanical Limited in the United States. That's a joint project we are working. So their problem is they are using river water from the Klamath Falls in Oregon State in USA. So it's because of algae bloom, the water is getting more contaminated. They came to us to help them with technology to protect them from this algae bloom. So we suggested them give us some sample, we'll test it, and we find out what kind of algae is present in that. If it is possible, we can cultivate that and make use of that. If it is a hazardous algae which produces toxic compounds, then the only solution is to use flocculant to flocculate it and then make the water very clear. So we did some trial runs with our flocculant, what we developed using organic sources. It gave a nice result. It also significantly removed the toxic compounds produced from this algae bloom. However, their solid percentage is around 2.5, which is a high concentrated solid to handle with the flocculant and also with the ultrafiltration system. Therefore, we need to dilute that and then flocculate it and remove it. The removed algae can be used as a fertilizer straight away. The plant can take it, but aquatic animals cannot handle that toxin.
Yes, sir. Sir, actually, can the future be like algae lamps alternative to any light energy? So like street lights in the nights, algae lamp. So one of the application.
It's already in some countries they are using it, especially the Finland. I have seen some algae related lamps. It's similar to a solar thing. It's already in existence, but in the near future, yes, definitely, it's going to be a big.
Thank you so much, sir.
Thank you, Professor Swaminathan, and thank you, Kavya, madam. Are there any other doubts? You may, if you have any clarifications needed, you can contact Professor Swaminathan through his mail ID also. He has shared, and with the permission of Professor Swaminathan, I suggest the participants to make use of him for your research works as well as for your students' examiners. Can we say that, Swaminathan sir?
Yes, madam, of course.
Yeah, you can use
But,
it
but
because
unfortunately, Anna University and other universities are not allowing Indian NRIs to be international examiners. But maybe if it is relaxed in deemed universities, then I'm happy to do that.
it's even in NIT, it is that facility is there, sir. Only in Anna University that is there. Maybe in future they may change it.
Yeah.
So I request all the participants to make use of Professor Swaminathan, who is very much helpful, and he says yes whenever we ask any help from him. So he's the right person to help all of us. So I request all of you to make use of him.
One small request.
There is a person, sir, somebody is asking question.
Ma'am, I'm Vignesh from Anna University, Technology Commercialization Officer.
Yeah, that's Sir Vignesh. He's Mr. Vignesh. He's an alumni of our Tiruchi campus. Swaminathan sir, Vignesh is asking some questions to you.
No, it's a request actually, sir. So I could see that the business with algae, right? So in case if any of the startups is interested to pursue entrepreneurship in related algae related business, will you be helpful in suggesting us the machineries or the setup that is required for scaling up? As such, I could find that the most of the research is in the laboratory scale level, but I could see that some of the pictures you pointed that I mean, it is scalable. I mean, the scope for scalable is high. So is that can you suggest us?
Swaminathan sir, before Vignesh, please introduce yourself elaborately. He's an employee of Center for Technology Development and Transfer
Yes,
in
sir.
Anna
So
University.
I'm actually sitting in the Center for Technology Development and Transfer in Anna University, and I'm appointed as a technology commercialization officer, wherein I mine all the technologies from Anna universities across all the engineering colleges, university colleges, as well as from Anna University campus. So this is
for your question, that is our major role for recent years. So I'm consulting many companies. So it's definitely possible because the first priority will go to my state, which is Tamil Nadu state. Don't worry about it.
Fine. I will just take your contact information from madam, and I will keep you posted. Sure.
Yeah, that's in one of my slides as well. You can get it straight away.
Sure. Thank you.
We have the next session shortly. So, Swaminathan sir, shall I ask the participants to mail you if there are any other queries?
Yes, madam. So I have provided my email ID. They can contact me anytime with their questions. I can answer them. I have also provided my number directly, so that's easy to contact. So they can take advantage of that as well. So thank you for this opportunity and patiently listening in that after lunch hour. So try to make use of this, and I'm happy to get collaborated. Thanks, madam, and thanks for all the participants.
Thank you, Swaminathan sir, for your excellent elaboration about algae treatment using algae. It was a very wonderful session because participants themselves have given their feedback in chat box. You have got a very good I think participants have enjoyed your presentation very well. So thanks for spending your valuable time with us. Thank you so much, Swaminathan sir. So participants, you can, there is a lunch session. You can join exactly by 2:30 in the afternoon. We have a very interesting lecture in the afternoon by Professor Babu from Vellore Institute of Technology. He is also a very experienced person. He will be sharing a nice presentation with us. Please do join with us by 2:30 after lunch, exactly. Thank you so much.